This question deals with the titration of a diprotic acid. Now, in the case of polyprotic acids or polyhydroxide bases, the important thing to remember is that these processes are sequential. For example, in the case of this diprotic acid, first the initial proton is removed, and then in a second step, the second proton is removed. The net result is that there are two buffer regions, and we see the two buffer regions here. This region here refers to excess base that has been added. Halfway to each equivalence point, we have the usual situation where pH is equal to pKa, but applied specifically to this diprotic acid, we see that pH 1 is going to be equal to pKa1, pH 2 is going to be equal to pKa2. Now, it's a little bit difficult to, this graph is small, and therefore we're going to have to do an estimate. But we can see the first equivalence point is about at 20 milliliters. That's where we see the sharp increase in pH. Halfway to equivalence is about 10, and if we draw our line over to pH, you see it's about 2. So with the choices that were given, we can actually answer the question now, but let's go ahead and confirm the second pKa. The second pKa is going to be about 40. That's where we have this next huge jump. So at about 30 is halfway to the equivalence point. It's somewhere about 6 or 7. So B is going to be our correct answer.